been already introduced, thank you very much. I'll be talking about text mining in the biomedical domain. I'm part of the OntoGene group at the University of Zurich, of which the head is Fabio Rinaldi, who was originally planning to do the talk himself, but he wasn't sure if we could make it in time because of some other commitment. So, yes, there we go. I will first give you a few, two use cases of text mining in biomedical literature. One is, I think you cannot read any of this, so maybe I'll just skip this and jump right to the motivation part. So, sorry about that. So, like in most research, knowledge is generated in experiments and then published in scientific literature, which is good for humans to read, but it's hard to process it with machines. So therefore, in the bottom you see all these acronyms and logos of different initiatives, which put a lot of effort into generating databases which contain facts, knowledge facts, important facts in machine-readable databases. So a part of this information is put into databases by the researchers themselves, but most often this has to be done manually by human experts in a cumbersome process which is called curation. And this is exactly the part where we want to help with text mining in order to make this more efficient and maybe also more enjoyable. Also, we're trying to help in the opposite direction. When performing research and when the researcher is in the need of information, we want to help the researchers in getting to that particular information more quickly, be it if information comes out of databases or out of the full-text literature. Just a short side note, this is an old graph, but it shows the number of publications in PubMed, which is a repository for biomedical literature. And as we heard earlier today, the pace of publications per 10 seconds was 0.8, so it is being set for only for PubMed, there are about 2 publications per minute. So it's like 40% of all of the publications. They really need help here. Sorry. There we go. This is a workflow scheme of a big project which is doing semi-automatic curation with a very broad coverage attempt, and it is called RegulonDB. It has a very specific focus. It's just one strain of E. coli bacteria. It is funded by the US NIH, but it's actually taking place at Mexico, and Fabio Rinaldi is one of the collaborators there. So what is the task? They are going for a regulatory network, a network of gene regulations, and part of the information is already there in a curated database. So we have kind of the nodes of the network are already known, but the interactions, the edges between the nodes are not yet known. So we need to find these. So it means one could just read through all of the articles once again and add this information. But the approach here that is taken here is using text mining in order to filter the articles and just present those sentences which has the highest probability of presenting the parts of information which are missing. In a proof of concept study early on in 2013, I think, before the project started, this was tried with human curators, and they were able to find of those regular interactions, the primary information they were interested in, they were able to find all of them in just the filtered sentences. And the secondary, the growth conditions, the secondary information they were also looking for, they were able to find 3/4 of those. And all that by reading only 10% of the articles. So it really speeds up the work here. The editor, the interface you just saw is called OntoGene. It was developed by our group. It is pretty lightweight. It runs in the browser and has been applied several times, like the bottom one you see it in that pilot study. The bottom right here, you just see some figures on how happy the curators were when using OntoGene for another curation task, but I will not go into details here. Rather, let's switch to the second use case that I'm presenting here, which is the detection of protein-protein interaction. In a collaboration with Roche, we built a search interface which enables users to search for protein, much like in an information retrieval setup where you put in protein names and then you get relevant documents. This is the inverse of the curation task where you are given one document and then you find therein the most important information and rank these. And now we're going from relevant entities to documents. Let's just have a look at the search interface. Here, this is the entry page. One can either enter the name of a protein in the top search field or just click one of those top ranked proteins which are listed here. We will now go for TP53, which is a protein plays an important role in cancer. We here now see a list of ranked protein partners. So those are the blue ones are partners that are seen as highly probable to show an interaction with TP53 given the textual evidence in the collection which we have processed. So when we now click on the top one, MDM2, then we get a list of documents, and out of every document there's a small list of snippets, and the proteins we're looking for are highlighted. So you can click on one of the documents and then you either go to the source of the document, PubMed, or you are brought to Odin, the editor which I showed you before, where you can work on annotation. Just quickly, I want to talk about competitions. There is a number of shared tasks in the domain of biomedical text mining, one of which is the biocreative shared task. Our group also has participated a number of times. For example, here participants were asked to find protein-protein interactions like I just showed you before. They were asked to find pairs of protein mentions, resolve them to their database identifiers, and rank them per document. They were given full documents with all images and tables and everything, and had to extract this. Well, one of the problems was actually to get to the plain text, because the documents were given either in a messy HTML, which contained a lot of boilerplate, or scanned PDFs with all the problems that entails, like OCR errors or bad layout recognition. There was a number of biocreative shared tasks that are regularly held, and one of them in which we participated was the CDD triage task. You see here that our group performed particularly well there in overall results, and also in some of the subtasks like detection of genes or detection of diseases. Yes, this is just a quick side note. We want to now look at the approach which our group is taking in this OntoGene approach. We're working. Why is this going further when I stop pushing? We are working on a dictionary-based approach, which means we're working with a list of terms that we are looking for in a document. So in a very simplified way to say that. Most of the time we're using some of the existing knowledge ontologies or terminology resources, like the ones you see here. The reason for using a dictionary-based approach is that most of the times we need both a mapping from the terms to the database identifiers, and there's a way around going through a list which gives you this information. We have recently compiled a huge resource with different entity types, like genes and proteins, chemicals, disease names, organism or species name, and also cell lines, which summed up to about 13 million entries, so that we can target these different types of entity at the same time when processing documents. We studied some statistical properties. It's quite interesting how they have different degrees of ambiguity or diversity, but I'll skip this for now. Let's look at the document, which here shows both chemicals in green and proteins in blue. Notice there's one case of a disease name, arthritis, in yellow below the middle. This stage of the process is called term annotation or term matching. This is only one part of the whole pipeline we have. This here is a schema, the workflow of interaction or relation extraction, so the process of not finding single entities, but also combining them pairwise into interaction candidates. And you can see here in the middle, in the middle you see domain entities. This is the term annotation step we just saw. This is preceded by some NLD pre-processing, like tokenization, of course, lemmatization, POS tagging, and not always, but in some cases we also did further processing, like chunking or even parsing, syntax parsing, and the final step is then the relation extraction. Talking about syntax, for some approaches it has been proven to be very useful to actually look at syntax trees. So for each sentence we built up a dependency structure of the whole sentence, and then with a set of rules we were looking for sentences with matched entities. So appropriately certain positions were found, and if such a rule fired, then the system could deduce that those two entities are participating in an interaction. So some highlights. These are shared tasks in which our group has participated and has got good results or even best results, some of that. But now finally I want to show you the most recent or current project we're working on. In the recently just finished collaboration with the veterinary faculty of the University of Bern, we were working towards the ultimate goal of building a health monitoring system for cattle and pigs, not so much poultry. And we were working with pathology text, with post-mortem reports. And we were looking for indicators of certain diseases. Unlike other projects we have worked with, there was no previously existing terminology resources for that particular domain of disease indicators for in the veterinary domain. So a lot of the time was spent in constructing such a terminology resource. You can see here a number of terms that, in German actually, since the text for almost all of them in German, which all point to more or less the same disease, namely the torsion of the big intestines. Some of them are more specific, some are a bit more general, and we also have variation like spelling variations and misspellings or inflectional variants. And part of that could be modeled automatically. A lot of the terminology construction was done by hand, but it was assisted, particularly in cases like here, spelling variation with automatic methods. In a second step, we try to enhance this terminology with means of existing ontologies. The ULS, which was just mentioned in the previous talk, is a meta resource which connects the knowledge of a lot of medical terminology resources, and by connecting to them with some fuzzy matching, we were able to find other terms in our text which were also possible indicators for the same diseases which we already have been looking at. The next project I quickly want to present here is the SciMine project, which is funded by the Cogito Foundation. Its main aim is to find potential causes of mental disorders in a very large collection of texts, so basically all of the biomedical literature if possible. And the current stage of the project, the manually curated corpus is being created. And finally, another project, which is called Melanobase, is once again targeting more or less the whole biomedical literature, but focusing on all relevant information in the domain of melanoma. That project has just only started, so cannot give you much more detail to that than what is stated here. So to sum up this very broad tour of the works of the OntoGene groups, we could show that text mining technology can be very helpful in doing assisted curation, which is a very important task in the biomedical domain. And particularly the editor interface Odin, which was created, is useful in that task. Our group OntoGene provides technologies which have been proven to be of high quality in the past. And the new projects which are currently running, we're hoping that we are able to repeat those promising results. Thank you very much.
Thank you very much, Lenz. You were actually afraid that you couldn't make it within 20 minutes, so I see that you were much faster. So we have plenty of time for questions then. Please go ahead.
You told you have a dictionary approach for text mining. How do you deal with ambiguities, or you don't?
A lot of the time we're doing interaction extraction. And a lot of the approaches we do not deal with ambiguity here at this stage, but only in the end when dealing with relation extraction. So basically for each term that we find, then we keep a list of possible resolving entities, and then at the stage of relation extraction, we try which of those will then be most possibly the interpretation which participates in the interaction. So it's a post hoc approach. Does that answer your question?
How do you evaluate your methodologies?
There are different approaches, but often, so if there's a ranking into it, like in the relation extraction, the thing we showed you for the Roche collaboration, we're using information retrieval measures like TAP-K, so threshold average precision, which is like you look at the number of, say, you look at relevant documents until you have found up to, say, 5 irrelevant documents when you stop evaluating. Well, it depends on the task. So, in order just to evaluate the term ledger here, you can use like just precision and recall in order to tell if it's finding the relevant things or producing too much.
So you have a manual baseline compare.
Yes, sure. So that's where you're going. Yes. So when task where we have a gold standard, then this is where we can do that. So it's a research project that's where we do it.
Questions?
We just have to get answer. I'm not Fabio Rinaldi, who was supposed not to be here, but just manager the last week. And so, just to add something to your question, yes, both standard corporate against which we can compare. One very well-known one is called Craft, which is in Colorado. But also participating to all these competitions is a way to evaluate, because in those competitions the organizers provide manually theoretical standards, and so in each of them you're evaluating an aspect of the system.
Questions? Your products, your tools, are they freely available?
I can't tell about the code that was being produced before I was here, but we haven't actually kind of published it. But
They are available upon request.
yeah.
Okay, then thank you very much again, Lenz, for your presentation.